This is a production of Cornell University.
So I'll start by demystifying the title a little bit. I won't reread it to you since Michael just read it. But essentially what this means is that there are individuals of Aspergillus flavus who produce aflatoxin and individuals that do not, and both are maintained through evolutionary time, which means that producing aflatoxin must confer a benefit to the fungus sometimes, and not producing the toxin must confer benefit at other times. So I'm going to just give you a little outline. I'm going to start with some background on secondary metabolites in general, moving into background on Aspergillus flavus and aflatoxins, talk a little bit about work we've done in looking at A. flavus population structure, talk about the hypotheses behind that we think might be driving balancing selection, and spoiler alert, we think it's insects and soil microbes, and then concluding. So secondary metabolites are generally defined as just any compound that's not required for growth. And interestingly, in fungi, they've been hugely diversified. And this is partially due to the diversification of polyketide synthetases and non-ribosomal peptides amongst peptide synthetases, amongst other things. But just because we know of all these compounds, it doesn't mean that we know what they do for the organisms that produce them. So for example, here in Magnaportho oryzae, it was discovered that melanin, which we normally think of as a UV protectant, is also required in Aprosoria for structural integrity so that it can actually punch through plant tissue. So this is to say, even when we think we know what compounds are for, there may be other functions, or we may just be wrong. So to start off with some things that we have a sense of what they do. These are host-specific toxins, which determine the host range. So you can imagine if it produces one of these compounds, then it's able to infect a new host, and that obviously confers a fitness advantage to the fungus, so it would be selected for. And with virulence factors, virulence is not the same thing necessarily as fitness. You can have increased virulence without increased fitness, but in general, it does correlate to some degree. So again, it suggests that we have a sense of what these compounds are doing. They're allowing the fungus to infect a new plant or to better infect a plant and thus reproduce more. But in the case of deoxynivalenol, which is also classified as a mycotoxin, it's known as vomit toxin, endearingly. It's again, the way that we find out about these compounds is because they are toxic. So patulin we heard a little bit about last week, so I had to throw it up there. But ergot alkaloids, even back in the 15th century when Bosch did this painting, you can see that St. Anthony's fire was an issue. Long before we knew that there were microbes causing these diseases, we knew about the compounds, well, we knew about the diseases they cause, and we've gone on to learn about that. But this is all from a human perspective instead of what it does for the fungus that produces the compound. And that's, of course, important because mycotoxins contaminate 25% of the world's food supplies, and they cause disease and stunting of growth and even death. But again, while that's sad and costly to everyone involved in agriculture, what I want to know is why do the organisms that produce these compounds produce them? And just to drive home that we really don't know what these compounds do. So we think about antibiotics. Antibiotics, if you ask anyone, why does a bacterium produce antibiotics? They'll say, to kill bacteria, or a fungus produce antibiotics, to kill bacteria. And we know that they're effective at killing bacteria in lab and medical settings. However, in their natural settings, so like in soils, the levels of antibiotics are actually sub-inhibitory. So the idea that they're killing bacteria has kind of gone out the window as far as antibiotics in their natural environment. Instead, people now believe that they're used for intraspecific communication in the soil environment. So this kind of brings home the idea that we have to think about not what these compounds are doing to us, but what these compounds are doing in the natural environment at the concentrations and in the context of the natural ecology of the organisms that produce them. So now moving on to the star of the show, aflatoxin. Aflatoxin is an incredibly important mycotoxin. It is the most carcinogenic naturally produced compound, which has been a great line from my elevator speech when I'm talking about my research. It affects the p53 gene, which regulates the cell cycle. So if it disrupts that, then the cells become aberrant and just grow out of control. It's especially associated with liver cancer, which is a particularly nasty one. As such, it's highly regulated by the FDA in a bunch of crops, including maize, cotton, peanuts, and a variety of tree nuts. Today, as you notice from the title, we're going to be looking at Aspergillus flavus, but it's actually produced, aflatoxins are produced by several species closely related to Aspergillus flavus. And here you can see Aspergillus flavus growing on this ear of corn. And you'll notice that it's a yellowish green color. That's going to be important later. I want you to retain that. And then this is an infected kernel fluorescing a bright blue. So aflatoxin, the one that we're most interested in, is called B1, is it fluoresces blue. So again, the colonies are a yellowish green, it fluoresces blue. And here we have Aspergillus flavus growing on a plate, and we're hitting it with UV light, and it's illuminating. The red is just it overloading the system. But what you can see here is that different isolates produce different amounts. So these two are not producing any. This one's producing, these two are producing a little, these are producing more, and these two are producing just a lot of aflatoxin. And what this illustrates is that only 40 to 60% of Aspergillus flavus isolates produce aflatoxin. And that people have looked at the genetic code. So this happens across the country, 40 to 60%, all over the place. But also, if you look at the genetic code, you find evidence of balancing selection, which means that this is a persistent polymorphism. Again, it means that at some point, producing aflatoxin confers a fitness advantage, and at some point, not producing aflatoxin confers a fitness advantage relative, at least, to the other chemotype. So we're really interested in this balancing selection. It's important to note that while balancing selection is rare for secondary metabolites, as far as we know, it's not unique. So trichothecines and Fusarium, Graminearum are also under balancing selection. But even here, this is a little bit different. So this balancing selection is for different chemical profiles as opposed to produce and not. So this is kind of an interesting system in addition to being agriculturally important. So my mantra today are, what are the ecological factors that are driving balancing selection? And as I've been saying, in order to get at this question, we have to look at the natural ecology of the fungus. So the fungus, the natural environment of the fungus is typically considered soil, where it produces conidiophores that disperse spores everywhere. And I really do mean everywhere. It has a global distribution. And because of the spores being distributed everywhere, it can get into crops while they're in the field. In maize, it infects the silk. It grows through the silk and then gets into the kernels. But it can also be a post-harvest issue because it's just in the air everywhere. Now, if we're trying to understand what aflatoxin is doing for it, we might expect aflatoxins only to be sequestered in the sclerotia as like overwintering structures to protect them. Unfortunately, it didn't give us any kind of sense of that, because the aflatoxins are everywhere in it. It's in the mycelium, it's in the conidia, it's in the sclerotia. And also, it's not as clear-cut as just being a virulence factor. It's not like those that produce aflatoxin can infect a plant, and those that don't can't. So no hints really on what aflatoxin is doing, but again, this is just to give some background on what the natural ecology is that we need to look at to understand what aflatoxin is doing. So if you can't just tell what aflatoxin is doing by where it sequesters it in the fungus itself, maybe there's global distribution patterns that can kind of start to help us understand. Now, aflatoxin has been known to be more common in hot, dry areas for a long time, but it's a little unclear if this has to do with the stress on the plant. So this plant gets stressed, it's easier to infect, you wind up with more aflatoxin contamination. Or if this is just a phenomenon where hotter temperatures at lower latitudes select for aflatoxigenic, while the cooler latitudes select for non-aflatoxigenic. And so when looking at soil here, Manabe in 1978 showed that the black portion here is aflatoxigenic, those that produce aflatoxin. As you can see, he showed that as you go farther and farther south, this portion is increasing, suggesting that at lower latitudes in hotter areas, there's more selection for aflatoxin production. So we decided to follow up on this because Japan is far away. We decided to do it in the United States. So we developed 2 transects. So these are roughly of the same latitude here across. And so we did sampling at all of these locations with the help of collaborators who scooped up soil and sent it to us. And then we isolated them on soil dilution plates. So this is a rose Bengal plate, and you can see the yellowish green colonies here are Aspergillus flavus coming up. So we take the soil, we put it onto a plate, and then we can isolate it with a single conidium. Also, these plates are just really cool looking. So what do we find? What we find is we don't see the same pattern as they saw in Japan 40 years ago. In the eastern transect, we don't see any real pattern at all. It's, there's a lower percentage of aflatoxin producers, it goes higher, it goes lower. If anything, in the western transect, it's going the opposite direction, where we're seeing more aflatoxin producers towards the north. This to say, there doesn't seem to really be a clear pattern. It's not as simple as just in southern latitudes, aflatoxin production is favored. So if it's not latitude, then what? Dan Jansen, who's an evolutionary biologist currently at Penn, wrote a paper that suggested that secondary metabolites in general, and specifically aflatoxins, may be an agent of interference competition, by which you produce these chemicals into a substrate and then you kind of claim the substrate as yours to consume, because nobody else wants to eat it because you've poisoned it, basically. Or interference competition can also be that the compound just keeps down other species. So he specifically suggests that this would be an agent of aflatoxin would be an agent of interference competition for insects and for soil microbes. Okay, so Dan Jansen suggested as an idea. Let's follow this up with a real hypothesis. So we started off with the hypothesis that aflatoxin production confers an advantage to A. flavus in the presence of soil microbes. Again, this would be through interference competition, where production in soil kind of carves out some resources for Aspergillus flavus to grow and have higher fitness. And the flip side here is that in the absence of these organisms, so in the absence of soil microbes, or in the absence of insects, we kind of assume a cost. So the production of aflatoxin takes energy, so we just kind of assume a cost. So this to say, while we're doing balancing selection, we're just doing one side of it with these hypotheses right now. I'll come back to that later. Okay, so we're starting with soil microbes. In order to look at soil microbes, we need some of the background. Aflatoxins are produced in soil, which is a good first step if you're hypothesizing that they have a role there. And aflatoxin contamination of soil is common, both in agriculture, where farmers have to bury really contaminated crops and the aflatoxins leach into the soil, but also just because detritus on the soil surface can leach aflatoxins into the soil. So people have found aflatoxins leaching into the soil and also produced in the soil. Now, aflatoxin does have limited toxicity to microbes, but in some studies, it does keep down some microbial communities. And interestingly, Bacillus and Aspergillus flavus have this relationship where Bacillus produces compounds that are very inhibitory to Aspergillus, and Aspergillus flavus produces aflatoxin, which seems to have the biggest effect on Bacillus species specifically of the microbes that people have tested. Okay, so to look at whether or not aflatoxin production confers a benefit in the soil environment, we took a falcon tube and we put soil in the bottom of it. Now this soil was either autoclaved or not autoclaved. We also tried gamma irradiating it, and the fungus didn't grow any differently in gamma irradiated soil from autoclaved, and autoclaving is a little bit easier. So we went with that. And then we either, in some experiments, we added toxin or we did not add toxin to the soil itself. And that was both to sterile soil or non-sterile soils. And then course, we have to add the fungus. So we added a bunch of isolates separately, either non-toxigenic or toxigenic isolates to these tubes under these conditions. And then we would extract the DNA and use qPCR to quantify the amount of DNA that was present. And so that's kind of a proxy for fitness. So the more DNA that's present, the more that it's growing, the higher the fitness. And this will be important later. My graphs are going to say relative fitness. qPCR, you quantify things relative to a standard curve. So relative fitness just means a measurement relative to a curve on here. You can just think of them as fitness numbers, but it'll say relative fitness. We also looked at HPLC to quantify the aflatoxins, make sure it was producing under some of our conditions, and make sure that our addition of aflatoxins actually happened. And then for a subset of the samples, we did amplicon sequencing to look at the microbial communities and try to understand what both the addition of aflatoxins and the ability of the fungus to produce aflatoxins, what effect that had, if any, on the microbial community. So again, to show you, this is what the microcosms look like. So these are the falcon tubes that I was talking about, and this is a close-up inside. So we added a little bit of maize, and here you can see a maize bit with Aspergillus flavus conidiaphores coming off of it. This is one of our non-autoclaved, so this would be considered natural soil. And you can see here there's a penicillium species coming up. We often see a whole bunch of different species that we haven't really identified, but I'll talk about some of the composition in a minute. So we did this initially. We looked at 3 different temperatures. So we had 25 natural and sterile, 37 natural and sterile, and 42 natural and sterile. And what you immediately notice is that the 42 is a stressful condition. Even under the sterile condition, the growth is way down. But at 37 and 25, the growth is more or less the same. These are not significantly different. These in sterile are significantly different. But we're interested in the question of what does aflatoxin do for it? Not does it grow differently in the sterile or at different temperatures? This was initially what we were doing because we didn't know what we were going to find. So we're like, let's look at all the temperatures. And what we find is that in the natural soil, we consistently see that the non-aflatoxigenic have higher fitness than the aflatoxigenic. So not producing aflatoxin seems to confer a benefit here. And this is only in the natural soils. If you look at the sterile soils, there's no clear pattern. Here, the non-aflatoxigenic has a little bit higher mean. Here, it's the aflatoxigenic. Here, they're exactly identical. But across the natural soils, there was a significant difference between the non-aflatoxigenic and the aflatoxigenic fitness. Okay, so this was the opposite of what we expected. We expected aflatoxin to keep down other soil microbes, allowing it to colonize more food and grow better. So we didn't believe this. This is experiment 1. We didn't really believe that this was a real thing. So we did it again. And this time we were like, we're going to hit it with a ton of aflatoxin. That way we'll definitely see the effect that we were expecting. So here we have an independent set of isolates grown again in natural and sterile conditions, natural and sterile, this time with the addition of aflatoxin in these 2 and no aflatoxin added here. And with this independent experiment that was at 37, so note that the best growth was at 37, so it was easiest to work with the fungus at that temperature. This is at 37. We see the same pattern that in natural soil, we see a significant difference between non-aflatoxigenic and aflatoxigenic isolates. Now, it looks like the same patterns here in sterile. This difference was not significant. This difference was. So we were, again, surprised by this. So we decided to do it a 3rd time. Now, what this graph is it's the 37-degree from the first experiment. So you've already seen this data. And the 37-degree natural without toxin added. Oh, also, adding the toxin, as you can see from the fact that these are the same, had no effect at all. Anyway, so this is from the previous graph. And then we did it a 3rd time with another independent set of isolates, and we got the same pattern. And across all of these, we again see a significant difference between aflatoxigenic and non-aflatoxigenic isolates. with the non-aflatoxigenic isolates having higher fitness. So to summarize, aflatoxigenic isolates have a fitness cost instead of the hypothesized fitness benefit. And this seems to only occur in natural soils. At least we only got significant differences in natural soils. And the addition of aflatoxin to the soils had no effect at all. So as we were really surprised by this, we had to ask, why is aflatoxin so costly to produce in soils? Well, the first piece of the puzzle, what we did was we added aflatoxin to soils and then we measured it over time. And what you can see is that, so here you have the dotted lines are sterile soils and the solid lines are non-natural soils at 3 different temperatures. And what you can see is even in the dotted sterile conditions, aflatoxins degrade at a really high rate. So it goes from about 2,000 parts per million aflatoxin in the soil to even a little over 1,000 in just about 4 days. So this is degrading aflatoxin really quickly. In fact, we think that this might be such quick degradation that it's difficult for the fungus to produce enough toxin to keep up with the rate at which it's degraded in soil. Okay, so this starts to paint a picture of maybe why aflatoxin is not a great thing, not great for fitness in a soil environment. Next, we wanted to look at the impact on microbial composition. So these are NMDS plots. Essentially, I don't want to get into it too much, but it's a multivariate analysis to look at the community structure. And what I want you to see here is that we have added toxin, and then we have the producing ability of the fungus. So added toxin, circles is no added toxin, triangles is added toxin, and then with blue and green, I forgot this. So the fungus is green, but it produces a toxin that fluoresces blue. So throughout the presentation, when there's blue, it's aflatoxigenic. When it's green, it's not producing the toxin that fluoresces blue, so it's non-aflatoxigenic. So green is not producing it by itself, blue is. And what you can see is that these points are just all on top of each other. There's no clear pattern. It's not like you get the producers going up this way and the non-producers going over this way. And again, this is the fungal community, and the bacterial community is even more so, just completely on top of each other. So this is kind of a visualization of the fact that we found no significant difference either with the addition of aflatoxin or with the ability to produce aflatoxin of the fungus itself on bacterial community, or sorry, on microbial communities. Now remember we had that, we had a specific idea that maybe bacillus would be the answer to our questions of why aflatoxin might confer a benefit, which it doesn't, in soil environments. So what this is it's demonstrating the log base 2-fold change. So these are actually small changes across all of the different phyla of bacteria that we found. And each spot here is an OTU, which for practical purpose you can think of as a species, essentially. So within each one of these, each dot represents a species of this phylum, and then how much it changed from its abundance from not having an aflatoxin producer, so a non-aflatoxigenic isolate, to having an aflatoxigenic isolate. So how much did it change in that? And what we find is that here in the firmicutes, which is where bacillus is, there's no significant difference between the prevalence of bacillus in microcosms inoculated with A. flavus that produces aflatoxin and those in non-aflatoxigenic microcosms. In fact, we didn't find any species, fungal or bacterial, that changed significantly between any of our treatments. So it really seems that the addition of aflatoxin and the aflatoxin producing ability has very little effect on soil microbes. So again, we found, instead of finding a fitness benefit to aflatoxin production, we found a fitness cost. Now remember at the beginning I was saying, for balancing selection to be maintained, you need the non-aflatoxigenic to be favored under some conditions and the aflatoxigenic to be favored under others. So here we're creating an environment in the soil when competing with microbes where not producing the toxin confers a benefit, essentially. And this may be because aflatoxin doesn't seem to have much effect in the soil environment. It's degraded quickly, as I showed you. and it doesn't seem to change the microbial community composition. And so, as I mentioned earlier, the most likely explanation for this is that in the soil environment that's low in nutrients and they're competing with other microbes, the cost, the metabolic cost of producing aflatoxin may result in a fitness loss. So this is the simplest explanation for why this is happening, why there would be the benefit to the isolates that don't produce the toxin. Okay, so our first hypothesis was about soil microbes. We did not find a benefit, we found a cost. We decided to try again. Maybe it'll have a benefit in the presence of insects. So insects have a known association with Aspergillus flavus. When there's insect infestations in the field, it creates openings where Aspergillus flavus can get in and infect in the field. Also in stored grain environments, it's known that insects like maize weevils can vector it. It can go through their system. It can come out in their frass. The spores survive, that is. And so as a result of this, their insects are often associated with increased aflatoxin contamination. And interestingly, aflatoxin is toxic to a large number of invertebrates in vitro, but none of these studies that have added aflatoxins to food have looked at what effect it has on fungal fitness. So they've just been poisoning insects and invertebrates. And this photo to show you, to kind of depict that anytime I go out for a walk in the woods, I always see some kind of invertebrate on a mushroom when I come across it. There seems to be a lot of this kind of interaction where invertebrates are trying to eat fungi. So we decided to look at whether or not aflatoxin production through interference competition would allow it to claim the resources that we give it better than its non-aflatoxigenic counterparts. So similarly, I'm going to start off with telling you the methods. Here we use little microcentrifuge tubes. We put a little bit of food in the bottom of it that both Drosophila larvae and the fungus could eat. Into the food, we either did or did not add aflatoxins, similar to the last time. And then we did or did not add larvae. So to a single microcentrifuge tube, we'd add up to 9 larvae. Then we either added an aflatoxigenic or non-aflatoxigenic isolate, very similar construction to the last set of experiments, and extracted DNA using qPCR as proxy for fitness again, and HPLC to quantify aflatoxins. So to give you a sense of what this looks like, here's the microcentrifuge tube, way zoomed in. Here's the food at the bottom, and what you can see here, the green is the fungus, as you will remember. And here on the sides are some Drosophila larvae that have started to pupate. And then here in the middle, this kind of white structure is a Drosophila larva that did not fare so well. It seems to have mostly died there, didn't make it to pupation. So the first thing that we need to figure out is aflatoxin toxic to our model organism? So over the course of 20 days, we raised Drosophila larvae on food containing increasing amounts of aflatoxin, and then we looked at the number of Drosophila larvae that pupated, and then the number that emerged as adults. And the takeaway from this is, yes, as aflatoxin concentration increases, the number that pupate and the number that emerges as adults goes way down. So aflatoxin is toxic to our model organism. But again, this is just poisoning insects. This doesn't tell us anything about the fungus and what we want to know about whether or not this aflatoxin confers an advantage. And just to bring home that it's toxic, here are Drosophila larvae in our model systems here and here. And here you can see the mouth parts. But these black dots in the middle are an immune response. So it's a melanization that's either due to a direct response to aflatoxin or tissue damage caused by aflatoxin that then elicits an immune response. So it really is toxic to our model organism. In case you were not convinced, this is now 4 days. So the last one was 20 days, which is why when we don't add the fungus, we don't see the drop off in number of living larvae. So number of living larvae over aflatoxin concentrations. Before what we saw over 20 days, it dropped off dramatically. Here in 4 days, they survive. You can tell in the tubes that they're not as happy, but they don't die within that span of time, unless the fungus is present. So here we inoculated non-aflatoxigenic, which is green, and aflatoxigenic, blue. And you can see that without any added toxin to the food, there's a significant difference in how many larvae survive living in the tube with the fungus, with the aflatoxigenic killing more of the larvae than the non-aflatoxigenic. And as you might expect, as you add aflatoxin to the tube, the benefit of aflatoxin decreases and they become more similar because now there's aflatoxin in both of these tubes where here there was no aflatoxin at all before. Okay, so again, this tells us nothing about our main hypothesis. This just shows that when grown with the fungus, the larvae die. So if we got into the fungus, here is the fitness of the fungus as measured by qPCR over increasing aflatoxin concentrations. And we have tubes that have larvae and tubes that do not have larvae. So the tubes that do not have larvae seem to have no change. In fact, there's no change statistically across the increasing aflatoxin concentrations. So just adding aflatoxin to the tube does not increase fitness of the fungus. However, in the presence of larvae, you can see with the 9 larvae competing with the fungus, there's a dramatic reduction in fungal biomass compared to no larvae. And this is ameliorated and becomes as the aflatoxin concentration increases. In fact, these last 2 points here at about 1.7 parts per million are not significantly different, whereas these two are about 20 times different. So adding aflatoxin to the medium clearly increases fungal fitness. However, we're interested in if the fungus's production of aflatoxin can benefit it. And here, the aflatoxigenic and non-aflatoxigenic isolates were not significantly different. You can even kind of see that they're just on top of each other throughout this experiment. And to kind of visualize what this difference looks like, here's a tube with no added aflatoxin, and you can see a bunch of the white food shining through. Here's 1.75 parts per million aflatoxin added to the food, and this lush green, yellow mat is Aspergillus flavus conidiating all over the surface. The larvae have all died, more or less. So before we were adding toxin to the tubes, we wanted to ask, does the natural production by the fungus itself have an effect? So here we just did non-aflatoxigenic and aflatoxigenic isolates without aflatoxin added to the food. And what we see is no significant difference between their growth when no larvae are present. But in the presence of larvae, we do see a significant difference with aflatoxigenic isolates having slightly higher fitness than non-aflatoxigenic isolates. But this is a little bit shocking. We went from having this kind of visualizable difference, 20-fold difference, to just kind of eking out a difference there. And so we had to ask, why is the difference so small? And what we have to remember is that Aspergillus flavus doesn't just produce aflatoxin. It also produces a bunch of toxic compounds like kojic acid, cyclopentazonic acid, and alpha-trem, all of which are known to be toxic to insects. And you have to remember that both the non-aflatoxigenic and the aflatoxigenic isolates may be producing these compounds as well. Also, the amount of aflatoxin that our isolates produced varied 1,000-fold, from 8 parts per billion to 8 parts per million. So caught up in this difference is a lot of the variation between isolates and what compounds they're producing, and even how much aflatoxin our aflatoxigenic isolates are producing. For context, 8 parts per billion, you could eat corn that was infected with that level in the US, not in Europe, but in the US it's regulated at 20 parts per billion. Okay, so there is a significant effect, a much smaller effect on fitness of producing aflatoxin, of the fungus producing aflatoxin in the microcosms. So what is going on in these microcosms? We said that this was an interference competition hypothesis by which it would claim resources. And what I'm going to show you here is there's a Drosophila larva. Here you can see its mouth parts, the little black parts there. And up here we have Aspergillus conidiophores coming off. can see that it reaches up to the conidiaphore and bites it. I'm going to play it twice more. Really gets its back into it. So what this tells us is that this competition isn't just competition. There's also fungivory going on. So it might be an interference competition story, but there might also be an anti-fungivory story going on here. We didn't wind up separating those. In fact, we kind of think that when the fungus is growing through a substrate, it might be inextricable. You can't separate what's fungivory and what's competition for resources because the fungus has grown through all of the resource. But what this does show us is that there's a direct eating of the fungus, which got us interested in what effect that might have on the fungus. And we were surprised to see that with larvae present, isolates produced on average, so this is aflatoxin production, no larvae added to the tubes, and simulated damage. So what I did was I took a toothpick twice a day for 4 days and stabbed each tube 30 times to simulate the kind of little mouth parts that Drosophila larvae would be eating the fungus with. And so with no larvae, they produce a baseline amount of aflatoxin. With a single larva, because if I added 9 larvae, it decreased the biomass so much that it was really difficult to compare between things. With a single larva, it increases aflatoxin. And when I stab it with a toothpick, it really does not like that. And what this shows us is that this is consistent with an induced resistance response, that it knows that it's being eaten, so it produces aflatoxin to defend itself. Now, it's important to note that because it also does this just when I stab it with a toothpick, just when it's physically damaged, not only when the larva does it, it's not clear that this is necessarily an evolved adaptive response. This could just be that when I damage it, it releases oxidative stress. Oxidative stress is known to increase aflatoxin production. And so this could just be that when you damage it, it produces more aflatoxin, not that it's an evolved relationship between the two. Okay? So to summarize, as we started out with, aflatoxin is toxic to our model organisms. Aflatoxin, when added to the medium, dramatically increases aflatoxin, or the fitness of Aspergillus flavus, but this is true for both tox and non-tox. It just shows us that it increases fungal fitness in the presence of insects, but not in their absence. And then we showed that the production of aflatoxin by the fungus itself, without us adding it, also increases fungal fitness, but if you recall, it was just a little bit. It wasn't nearly as much. And then finally, the graph that I was just showing you, aflatoxin production is increased when the fungus is physically damaged, consistent with an induced resistance response. So with insects, balancing selection could also be maintained by the variability in how susceptible insects are to it. So we tried similar experiments with maize weevils, and they were resistant to aflatoxin up to 40 times the concentration we used for Drosophila, so we just gave up at some point. They didn't care. So you can imagine if you're producing aflatoxin and there's a resistant insect, it might not confer the benefit that it does if there's a susceptible one. And although we were unable to show a fitness cost in the second experiment, we think that has to do with the nutrient richness of the medium rather than that there isn't a cost to it. So we saw it in the soils, but not here. So what does this mean for balancing selection overall? So if you have a piece of organic matter on the soil surface, fungus comes, the non-aflatoxigenic isolates will do better. So non-aflatoxigenic are favored when competing with soil microbes for resources. When competing with insects, however, the production of aflatoxin benefits and aflatoxigenic isolate individuals are favored. When you have everything going on all at once, we have no idea. It really depends on how strong these factors are in the field. And so this does not preclude aflatoxin from having other functions in other systems, and it needs to be field tested. So that's what I mean by we don't know what the relative strength of these factors are. So if there's both insects and soil microbes present, what's going on? And we've emphasized the potential for insect control to be good for aflatoxin reductions, because while it was known that insects vectored Aspergillus flavus before, it was not known that they could actually increase the amount of toxin that an individual would produce. And most importantly for our questions, this presents a conceptual framework that could explain what maintains balancing selection for aflatoxin production in Aspergillus flavus. And with that, I'd like to thank all of the people who helped us collect soil, with whom we couldn't have gotten the isolates. My committee members, Brian Lazaro and Eric Nelson, funding from USD AFRI, and my lab members and other collaborators on these projects. And with that, I would take any questions.
Questions for Nikki?
So what are the molecular underpinnings of being able to produce versus not produce aflatoxin, and how selection is executed at the molecular level in terms of, is this through recombination?
Yeah, so the question was, what are the molecular underpinnings of being able to produce aflatoxin or not being able to produce aflatoxin, and what effect that might have? So the aflatoxin gene cluster is composed of 25 genes, and the ways that non-aflatoxigenic isolates become non-aflatoxigenic varies a lot. There's some consistency, but it can go from a single nucleotide polymorphism to missing the entire cluster, and in just about everything in between. As to what effect having many of the genes versus missing the whole cluster has on fitness, we don't know. We've just found out that producing the toxin versus not has this effect. It's a little unclear if there's a difference between retaining some of the genes or not in the cluster. Andy?
So following up on that, you're comparing two strains that are genetically different. One can produce and one cannot produce, but how do you kind of deal with the challenge of any other genetic differences that are present in two strains? So
none of these experiments oh, sorry. The question was, if we're comparing one aflatoxigenic and one aflatoxigenic, one non-aflatoxigenic, how do we get at the diversity that might underpin differences between the two groups in the field? And so the answer to that is that we've sampled field isolates, and then in these experiments, we've used more than one isolate. So each experiment, I think, had a minimum of 4 or 5 of each type of isolate in it. So we hope with that we've captured some of the diversity and seen that.
I'm interpreting this as the aflatoxin is much more stable in the food substrate than it is in soil, even sterile soil. Do you have any explanations for that?
So the question was, why might aflatoxin be more stable in food than in soil? And I don't have a great answer for that. Soil's a really nasty place. I mean, it's known that humic acids and things and all of the compounds that are present in soils make it really difficult for compounds to persist there. But I don't know specifically what could be happening compared to in foods.
About microbes?
Well, it could be, but it was even in the sterile soils, so. Yep.
If aflatoxin is such a toxic material, do you have to exercise special techniques to protect yourself and others in the lab from possible toxicity?
Yeah, we followed, we use a biosafety cabinet when working with the fungus. We use gloves. The real issue with aflatoxin is when it's ingested. So I was able to refrain from eating any contaminated substances, but we still were careful with it, working with it in a biosafety cabinet and using gloves whenever using the pure aflatoxin.
Did the FDA inspect you at the lab?
Somebody did. Maybe it was Cornell. I don't know. It was EH&S did. They've been all over us. Oh, there we go.
Nikki, do you think with it was a corn borer was the second insect you tried? Do you think that insects that are more likely to come in contact with Aspergillus would have evolved some aflatoxin resistance compared to the Drosophila larvae, or do you think just larvae are susceptible to stuff?
So it was the maze weevil, but
Okay.
Yeah. So the question is, might insects that are more likely to interact with Aspergillus in the field more likely to be resistant to aflatoxin? And I wouldn't be surprised if that were the case. Seems to be a lot of variation in how susceptible they are. I know that the corn armyworm is susceptible to aflatoxin within the range that we were looking at with Drosophila, and there are a few other agriculturally relevant organisms that are susceptible to aflatoxin within the range that we looked at. But they haven't been done with the fungus there, so we don't know yet.
Thank you.
Okay, let's thank Nikki for a great seminar.
This has been a production of Cornell University, on the web at cornell.edu.